Hello guys, I want to make sure that you have all the tools and the resources that you need in order to understand the 4 types of irregular genetics that are important. And the first one we're going to talk about is the sex-linked traits, which are linked to the X chromosome. And if you remember, females have 2 copies of the X chromosome, and males have 1 X and 1 Y. So these traits are located on these specific chromosomes. Remember, humans have 23 pairs of technically identical chromosomes with 2 copies of each trait on each chromosome. However, males only have 1 X chromosome and a Y chromosome. The Y chromosome does not match up with that X chromosome, so it makes their genetics a little bit different. Females have 2 copies, so it's almost just like any other chromosome pair, like the pair 1, pair 2, or pair 3, where you could find the matching allele or matching gene for each trait on the other chromosome, but this one only gets 1 copy in the male. So this is a male chromosome. That's the sex chromosomes for male, and this is the chromosomes for female. And that's kind of important for understanding this. So the notes for this say, in humans, hemophilia B is a recessive trait. So that's really important. So remember, recessive, the only way that a recessive trait will show up is if there's 2 copies of the recessive allele, which we call homozygous recessive. So in order to have hemophilia B, if this is regular genetics, so here's hemophilia B. In order to have it, you would have, let's use the letter H. In order to have it, if it was not a sex-linked trait, you'd have to have a homozygous recessive genotype, okay? Now that's true for females because each copy has 1 trait on it, but males only have 1 X. So in order to have hemophilia B, really their genotype for a sex-linked trait looks like that. It's a lowercase h. Okay, so let's start with this. Let me go through it. The disease does not allow the blood to clot. So when you get cut, people that have hemophilia B, their bleeding does not stop on its own. They actually have to seek medical attention, and they take a regular medication to help build the cells that will help with blood clotting. And so during excessive bleeding can occur during trauma. So like a car accident, even a bruise can actually be something that's really, really dangerous. So we're going to use a letter H, or capital letter H, to represent normal dominant genes. So people who do not have hemophilia have at least 1 copy of the dominant allele for not having hemophilia. Okay, and those that have the disorder have 2 copies of the H, the lowercase h allele. So capital H will be normal, lowercase h will represent the gene for hemophilia, it's recessive. Since this is a sex-linked trait, you must show the sex chromosome of each parent along with the gene on each X chromosome. So it looks kind of funky, but I'll show that for you. It says, work out the following crosses and write the possible genotypes and phenotypes of the children. If a male has just 1 X, little h chromosome, then he has hemophilia B. A female must have 2 X little h to have the disease. So for males, here's their chromosomes, or sorry, for females, here's what you're gonna have to write. And I like to write the female kind of on the side when we do the Punnett square, because we're gonna do a cross between a female, a mother who is a carrier, so she has a dominant allele for hemophilia, or for not having hemophilia, and then one gene for the carrier. The male is completely normal, so you can see that the H is located on that X chromosome. There is nothing on the Y. However, you still need to write the letter Y in order for it to make sense. So you write X. So I'm going to put the X chromosomes on the side. That's the female. And I'm going to use these H's and attach them up here. They're a superscript, kind of like you would write exponents. But really, when you're looking at this, you're looking at the H's to determine the genotype, but the X's need to be there because we're talking about things that are found on the X chromosome. And then the male is XY, so here's his X and here's his Y. Remember, we leave the Y alone. There's nothing on that Y that we are going to care about right now. And we'll put his capital H on the X. So we're going to do the cross. So you do the chromosomes inside. I like to actually do the chromosomes first because it just makes a little more methodical process. Okay. And then now I'm going to attach the H's. So here's the cross for this H. I'll put the capital H here. Still, you want the dominant allele first. So I bring this H, capital H, to this X, this lowercase h to that. And then this H here, and nothing on the Y. And then I take this little H and put it over here. So these are all females because they have 2 X chromosomes, okay? So female, we have X big H, X big H, which is normal. They have a normal phenotype. They do not have hemophilia. We have a female who is X big H, X little h. She's normal, but she's a carrier. Okay, she's a carrier because she has that little h. So when she has children, or if she has children, she has a 50-50 chance of passing on this gene, passing on this gene, so she's a carrier for it, but she does not show hemophilia. We have 2 possible males. One male could be X big H Y, and since they have the big H there, this would be a normal, okay, and a male X little h Y, and unfortunately this means that little h means that this would be a hemophilia B male. So this male would have hemophilia. So if they have a boy, they have a 50% chance of having one with hemophilia. And so those X chromosomes actually get contributed to by the mother. The mother contributes those. So the dad, if they have boys, can only contribute the Y, and the X's come from the females. Okay, and the X's only get contributed by the male when there's a girl, when they have a girl. All right, hopefully this makes sense. Let me go through the rest of this. All right, the mother is normal, completely normal. She's not even a carrier. So her genotype is X, big H, X, big H. And then the father has hemophilia. He's X, little h, and Y. So X here and the Y here, but he has it. So he's just like the male up here. All right, so he has it. Now, they're going to have children and the possibilities of what they can have. Big H, X, little h. So these are females. The X's mean they're females, and they do not have hemophilia because they both have dominant alleles, but they're both carriers. So there's a 100% chance of the girls being born with this genotype for hemophilia, and 100% of the girls will not have hemophilia. So over here, X big H Y, X big H Y. So these are males over here. And none of the males will have hemophilia because the mother was completely normal. She wasn't even a carrier, and she contributed that dominant allele to her sons, so 100% of boys and girls will be no hemophilia. No hemophilia B, so they will be fine. But 100% of the girls will be carriers. 100% of females are carriers, which means they carry one copy of the gene. All right. Last one. Okay. Red-green color blindness is another sex-linked trait. It's just like hemophilia, but it's for a different trait. It's also found on the X chromosome, which means that you have to have 2 copies of the allele for females to show it, and unfortunately for the male, you only need 1 copy. That's why some of these traits, like hemophilia B and color blindness, are more common in males than they are in females, just because the males only need like 1 copy of the allele, whereas females have potentially 2 copies of it. So females can carry it, males cannot. They either have it or they don't. Red-green color blindness comes from the development of certain rods and cones in the eyeball and makes it so that you can't tell the difference between red and green colors. They still see colors. It's not like they see in black and white, but they won't be able to tell if it's red or green. So anyway, we're going to use the capital C to represent the dominant gene for normal color vision and little c to represent the gene for red-green color blindness. So it says, show a cross between a man who is colorblind. So here's the male. If you don't know and you're trying to figure it out, it's best just to kind of write down the genotype. So show a cross between a man who is colorblind. Here's the male. So he's going to have a little c. Make sure it's little. I know c's are sometimes hard to tell because they're the same shape. and a normal woman who is not even a carrier. So she's going to have a large C on both of those X's. So you need to do the crossing, X, big C, X, little c, X, big C, X, little c, X, big C, Y, X, big C, Y. So we have of the girls, none are colorblind. Of the males, none are colorblind because they got the normal gene from mom. Dad is a carrier. Dad has it, so he's going to pass it on to his girls, but their girls are going to be carriers. So it says, can they have a colorblind daughter? So the daughters are here. They're XX. No, they cannot because they have one copy of the dominant normal gene. Okay. Can they have a colorblind son? I just said that. No, they can't because they both have copies of the dominant allele. So no, they cannot have a colorblind son. Can they produce daughters who are carriers? So here's the daughters. And yes, 100% of the daughters are carriers. So yes, 100% carriers because the dad is going to give them the little c. All right, hopefully this helps. This is the sex-linked or X-linked traits. They follow regular Mendelian rules where you have dominant and recessive, but it's found on the X chromosome. And remember, males only have one X chromosome and they have a Y, so that's why it's not quite regular genetics, because in regular genetics, the cross is between two alleles for both parents.